for kind introduction. It's a really big honor to me to introduce my research for I did during my postdoc, and I really like the we have non-sugar drinks for today because I'm gonna talk about the liver disease induced by sugar intake. And then, so I'm gonna talk about how how eating salad or like vegetable can treat the NAFLD, like fatty liver. So MESUD, one of fatty liver disease, metabolic dysfunction associated steatotic liver disease, is most likely the growing indicator for liver fatality. Sorry about that. Can I move this? Oh, okay. Come here. Ibarra. Okay. I'm sorry.
By clicking on the screen.
Freak. Oh, okay. Let me try. Okay. So the excessive consumption of sugar, which is high fructose syrup, as liquid form, like soda or juice, is positively correlated with the fatty liver. Those toxic features with is it related with its catabolism. So first, when we drink soda, fructose can be catabolized by intestine, and excessive fructose can be spilled over to colony microbiome, and it produces a lot of the short-chain fatty acid here. Okay, and then once fructose reaches to liver, it acts like transcriptional activator for lipid synthesis in the liver, which is de novo lipogenesis. And also the short-chain fatty acid with mostly acetate can contribute this hepatic lipid synthesis. So like high fructose syrup, which composed by one glucose and fructose, is known to the bad effect to the body, like metabolic disease. However, the inulin, which is monofiber and soluble fiber, is composed by polymer of fructose, and it's known to as some good effect, like beneficial effect or prevent metabolic disease. Then here, the question arises first, if high fructose syrup, which is fructose, and inulin are consumed together, which one would win? And it's metabolism. So to determine the effect of inulin on metabolic disease induced by fructose, we generate 5-year-old mice like this. So first, we fed mice control that with water, inulin diet with water, and control that with high fructose syrup water, inulin diet with high fructose syrup water. And we added one more group, the last one, to see the inulin, if inulin has the treatment effect on metabolic disease. So first, we fed control diet with high fructose syrup on mice, like first 16 weeks. And then we switch inulin diet, but then we continuously feeding the high fructose syrup water and to see how inulin like changes fructose metabolism. So this is most striking phenotype that induced by those dietary intervention. So you can see here, those three groups, the consume high fructose syrup water, but only the control diet has more fat accumulation in the liver. But you can see here the inulin diet or delayed inulin diet, very much less fat accumulation. And further, we analyzed lipidomics of liver sample. And then, as you can see here, we could detect the global reduction of various lipid species in the liver. And also, we found that liver fibrosis marker gene expression also suppressed by inulin supplementation. Okay. But one paper published at the big paper published the major contribute fatty liver or like fatty liver disease is de novo lipogenesis. So fat synthesis in the liver is major contribute to induce the fatty liver disease. So next, to determine the inulin effect on the lipogenesis, we perform isotracing using D2O. I'm sorry. So first, we found that 14 weeks of high fructose syrup drinking. Okay, as you can see in the graph, when we compare the orange and the red, the high fructose syrup drinking induced lipogenesis. And when you compare the red and then green, the inulin supplementation suppressed it. After we switched to the from control diet to inulin diet, we surprisingly found that inulin diet suppressed lipogenesis even 2 weeks later of inulin diet. And it maintained the low lipogenesis, even mice was drinking high fructose water. Not only lipogenesis, next we analyzed fatty acid oxidation. Since this here, malonyl-CoA is one of the intermediate of DNL, can suppress fatty liver oxidation. So we measure the labeled acylcarnitine and labeled fatty acid in the liver, and we calculate the ratio, which show like the ratio of substrate and product. So here, inulin increased this ratio, which indicate inulin increased fatty acid oxidation in the liver. And we did another experiment. We orally treated label palmitate to mice and then measured the label CO2 gas using indirect calorimetry. And as you can see here, higher green, the inulin enhanced fatty acid oxidation. So, so now we wonder how inulin supplementation suppress hepatic fat accumulation. So as I mentioned before, those 3 steps are major fructose catabolism. First gonna be by intestine, second gonna be by gut microbiome, third gonna be by liver. So let's check one by one. So first would be intestine, and then we found that inulin do not affect intestinal fructose catabolism by measuring gene expression level and isotracing. And next to, oh, I'm sorry. Ooh. Oh. Next, let's move on microbiome. Okay, so track the microbiome fructose catabolism. We use isotracing of the fructose. So we orally treatment label fructose, and then we quantify the fructose level in intestinal content. And then when you see the jejunal and ileum, we didn't detect any differences like in urinary supplementation. But here, cecum, like we detect the completely absence of label fructose in the inulin fed mice. Consistent with previous result, the label short-chain fatty acid, which is the major product of gut microbiome fructose catabolism, was similar depletion in the inulin-fed mice. But surprisingly, we could detect much higher label short-chain fatty acid in jejunal content, which is a part of small intestine. So this means inulin strikingly stimulate fructose catabolism of small intestine by gut microbial fructose catabolism. So next, to directly determine whether these whole effects are from the microbiome action, we treated antibiotics to the inulin-fed mice. So we added antibiotic cocktail to the high fruct construct drinking water to deplete the microbiome, and then we perform isotracing. Here, as you can see, the red bar, the antibiotics increase the DNL, which means antibiotics reverse the inulin effect. And next, we did secret content transplantation to see the microbiome's effect. So there is some two donor group, one of control diet with high fructose water, and one of the inulin diet and high fructose water. And we collect the secret content from those donor groups, and then we orally deliver the secret content solution to recipient group, and recipient group only fed control diet. And we found that this secret content transplantation block the fructose spillover to the colon, even though these recipient mice have not eaten the inulin diet. Also, the label, the fatty acid, which can indicate the lipogenesis level, is suppressed by inulin supplementation. Okay, so next we wonder then what happened in the liver tissue, since when we think about the inulin strongly suppress fat accumulation in the liver, then where those fructose carbon go? So next we use the fructose, the isotope, and then analyze the label metabolites. And surprisingly, you can see here, we could detect highly increased label glycine and serum level in the liver. And also using unbiased correlation analysis, we could find this increased serum level are strongly positive correlation with glutathione synthesis metabolite. So we also found not only glutathione, this metabolite also increase the level by inulin supplementation. And not only serine or glycine, one of the building blocks of glutathione, the cysteine, also important to synthesis, also important to synthesis glutathione. And we found the highly increased the liver cysteine transporter. And next, we use cysteine tracer and then found the label, the glutathione also increase in inulin-fed mice. And as you guys know, glutathione is antioxidant. And then finally, we think this increased glutathione suppress fructose-induced hepatic oxidative stress by measuring the oxidative stress marker. So if I summarize those findings at one slide, so intestine didn't change any fructose catabolism, and then inulin increased the jejunal microbial fructose catabolism, and it results in decreased colonic fructose spillover, and also inulin supplementation decreased hepatic oxidative stress. And those results finally that eventually decrease the lipid synthesis in the liver. Then here then we wonder which microbiome species mediate this inulin effect. So we perform 16S RNA sequencing of CCAR content and jejunal content of the mice. And then we found that those microbial community reveal significant differences among those groups. But among this identify these many bacteria, we want to narrow like species bacteria to produce inulin effect. So using unbiased correlation analysis, we narrow down 2 different bacteria candidates, which have negative correlation with liver fat synthesis and positive correlation liver serine production. So left is one of candidate bacteria that's anxiety-functious. I will say BA bacteria. So BA bacteria was increased their intensity in jejunal and CCAR content by inulin supplementation. But we found BP bacteria, which is Bifidobacterium pseudosyngum. It increased jejunal intensity, but it was not in CCAR content. So we wonder to determine this bacteria effect. This bacteria can reproduce inulin effect. We did bacteria colonization experiment. So we cultured single bacteria and then orally delivered those bacteria to 4 weeks, and we did tracing to see fructose metabolism. So first, we found that treatment of BA bacteria decreased the lipid synthesis, but we didn't see the same effect by BP bacteria. And also, labeled short-chain fatty acid was increased by BA bacteria, but it was not by BP bacteria. So this is conclusion slide. Dietary inulin supplementation decreases lipogenesis, and inulin promotes the small intestinal microbiome to break down fructose and block fructose spillover to the colon. Inulin supplementation activates hepatic de novo serial synthesis and cysteine uptake, which protect liver from fructose-induced oxidative stress. And we found that bacterial death anxiety-functions is the key contributor. I'm really thankful to all the members in Jung lab, especially Sun Jung, who supervised all this study. And I also thank to my funding source, ADA and NRF. Thank you so much. And then I will be happy to get a lot of comments from this study.
Questions for me? Can you give a little bit details about the animal experiment regarding the time course? Like how much was the infusion time and what of the sampling time? Because metabolism is affected by that. And also if you can say a little bit about your mass spectrometry and data analysis method.
Sorry, what was the second question?
Regarding your analytical platform.
Oh, I see. So we treated antibiotics for 2 weeks. We didn't any infusion. So we added antibiotics in the drinking water. So mice keep drinking this antibiotics solution. And then we treated 1 week to deplete all like random microbiome in the mice. And then we start the antibiotics, and then we like continuously doing for 16 weeks to induce lipogenesis.
But the fructose was there for all the time? 30
Yes, fructose antibiotics.
And then the deuterium exchange, which happens because you get the deuterium exchange happens after drinking. Did you account for that?
Oh, I replace every 2 days, make a fresh antibiotics. And then for metabolomics analysis, we are using Healy, using like the Sportos amide column. then like QE, a thermal QE plus model. Using that, we analyze metabolomics, and then as you can see here, we lipidomics. That is our typical tool for this method.
And regarding data analysis, how do you analyze data?
Oh, so we are using Maven software, yes, to like because most of my study is isotracing. So I think Maven is good for track the label metabolite. So most of we are doing using Maven, but some of untargeted or library searching we are using compound explorer like thermal software.
Any other questions?
Yeah. Thanks for the overview. For the role of the bacteria, that you have two species that were associated to the protective role of against the fructose related oxidation. And this data coming from mice. Probably the next steps will be to see if this is consistent in human data. Is this the next step, or you already have an overview of if that is the consistency that we are seeing?
Yeah, there will be great questions. But so there's many papers that already published about the effect of bifidobacterium, one of the both candidate bacteria. About they have improved effect to the diabetes or like metabolic disease, and also fatty liver. So many studies suggest this one has a beneficial effect, but they don't suggest like exact mechanism. So we suggest our mechanism, and then maybe next gonna be like found which microbiome, which metabolite, like a microbiome product can induce these changes. Then we can adjust those molecules to the human.
One more question.
So I was interested in your 13C palmitate experiment. Do you, because we've been trying to do some 13C palmitate, not very successfully. So what do you use to resuspend your palmitate?
Oh, we use soybean oil.
Okay.
Yeah, we pilot soybean oil. That was powder, right? So we use soybean oil and like increase like 30 Celsius to like better the powder.
All right, let's thank Sunhee one more time for her.